Hi, hi everyone. This is Suma. My topic is reverse transcribing virus. Let's move to introduction. A reverse transcribing virus is any virus which replicates using reverse transcription, the formation of DNA from an RNA template. Both group 6 and group 7 virus fall under this category. As we all know, the Baltimore classification of virus, under this classification, the group 6 and group 7 virus fall under this category. Group 6 contains single-stranded RNA viruses which use a DNA intermediate to replicate, whereas the group 7 contains double-stranded DNA viruses which use an RNA intermediate during genome replication. Next comes to group 6 virus. All the members of group 6 use virally encoded reverse transcriptase and RNA-dependent DNA polymerase to produce DNA from the initial virion RNA genome. This DNA is often integrated into the host genome, as in the case of retroviruses and pseudoviruses, where it is replicated and transcribed by the host. The group 6 viruses includes the family METAVIRIDAE, family PSUEDOVIRIDAE, and family RETROVIRIDAE. First comes to family METAVIRIDAE. Its typical members are Saccharomyces cerevisiae TY3 virus, species Saccharomyces cerevisiae TY3 virus, genus Metavirus. They are icosahedral in shape and might be enveloped. It has two identical copies of linear single-stranded positive sense RNA. Its replication by reverse transcription primed with a host encoded tRNA. Genomic RNA is translated into one or more polyproteins. Fungi, plants, and animals are its host range. Erantivirus and metavirus, including this species. Then comes to family Pseudoviridae. Pseudoviridae is unofficially classified under the group 6 RNA transcribing viruses and infect fungi and invertebrates. The viral capsid is unenveloped and looks roughly spherical. The capsid is round with icosahedral symmetry with triangulation number and 4. It is isometric to quasi-isometric and has a diameter of 30 to 50 nanometer. LTR retrotransposons are poorly characterized and the lipids have not reported. Genome replication takes place in the host cytoplasm or the nucleus, and assembly can occur in the cytoplasm or in the nucleus. Example, Saccharomyces cerevisiae TY1 virus, species Saccharomyces cerevisiae TY1 virus, genus pseudovirus. Next comes to family Retroviridae. They are positive stranded enveloped RNA viruses that reverse transcribe their RNA into a DNA intermediate during viral replication, hence the name retrovirus. The virions are spherical, enveloped, and are 80 to 100 nanometer in diameter. Glycoprotein surface projections are about 8 nanometer in length, and the internal core constitutes the viral nucleoplasmid. The virions are relatively resistant to UV light, and the surface glycoproteins may be partially removed by proteolytic enzymes. The RNA constitutes about 2% of the virion dry weight, and the monomers are held together by hydrogen bonds. Protein constitutes about 60% of the virion dry weight, and there are 3 to 6 internal non-glycosylated structural proteins encoded by the GAG gene. GAG gene means group of specific antigen is the major structural protein and comprises about 50% of the mass of the virus. Next come to retroviral infection. As we all know that retrovirus includes the HIV virus that cause human infections. The retroviridae constitute a large family of viruses that predominantly infect both human and animal vertebrates. The retroviral infections can cause a wide spectrum of diseases ranging from malignancies to immune deficiencies and neurologic disorders. However, Most retroviral infections occur without any detectable deleterious effect to the host. The retrovirus known as human immunodeficiency syndrome, as I said, it causes AIDS in the humans. HIV is closely related to simian immunodeficiency virus SIV, a retrovirus found in chimpanzees and gorillas. HTLV first was the first human retrovirus to be discovered, having been detected and isolated in 1979 by American virologist Robert C. Gallo and colleagues. HIV was first isolated in the year 1983. Next come to group 7 virus. Group 7 virus includes the two families, namely family HEPADNAVIRIDAE and family CAULIMOVIRIDAE. Both families have DNA genomes contained within the invading virus particles. The DNA genome is transcribed into both mRNA for use as a transcript in protein synthesis and pre-genomic RNA for use as the template during genome replication. Virally encoded reverse transcriptase uses the pre-genomic RNA as a template for the creation of genomic DNA. Example for group 7 viruses are family Hepadnaviridae, which includes a virus namely hepatitis B virus, family Caulimoviridae, it includes a virus namely cauliflower mosaic virus. First comes to family Hepadnaviridae. Hepadnaviruses have very small genomes of partially double-stranded, partially single-stranded circular DNA. The genome consists of two uneven strands of DNA. One has a negative sense orientation and the other shortest strand has a positive sense orientation. As it is a group 7 virus, replication involves an RNA intermediate. Hepadna viruses have a peculiar mode of replication. They replicate through an RNA intermediate. The three main open reading frames are encoded. Then hepatitis B virus. Hepatitis B virus causes serious liver infections. Symptoms of hepatitis B disease are abdominal pain, dark urine, fever, joint pain, loss of appetite, nausea and vomiting, weakness and fatigue, jaundice. Then comes to causes. What are the causes of hepatitis B disease? Sexual contact with an infected person, sharing of needles, accidental needle sticks, infected mother to child. And then comes to treatment by through vaccine only we can prevent the hepatitis B disease. Then comes to family Caulimoviridae. Caulimoviridae is a family of viruses which infecting plants. There are 94 species in this family assigned to 11 genera. The family Caulimoviridae are termed double-stranded DNA reverse transcribing viruses. It contains a reverse transcription stage in its replication cycle. This family contains all plant viruses with a double-stranded DNA genome that have a reverse transcribing phase in their life cycle. Then comes to cauliflower mosaic virus. They are pararetrovirus that infect plants of the family Brassicaceae. Brassicaceae family includes the plants like cabbage, broccoli, Brussels sprouts, and Solanaceae species. That Solanaceae species includes the plants like potato, tomato, all peppers, ground cherries, that is tomatillo, and eggplant. Eggplant. Gag gene. Gag Cauliflower mosaic virus induces a variety of symptoms such as mosaic necrotic lesions on the leaf surfaces, stunted growth, and deformation of the overall plant structure. The symptoms exhibited vary depending on the viral strain, host ecotype, and environmental conditions. Cauliflower mosaic virus is transmitted in a non-circulatory manner by aphid species such as Mysis persicea. Once introduced within a plant host cell, virions migrate to the nuclear envelope of the plant cell. Thank you, sir, providing me an opportunity to give the information about reverse transcribing
Thank you,
virus.
Suma.